Monocytes start to differentiate into macrophages. This bigger cell can eat a larger amount of bacteria than other phagocyte cells, but can also release cytokines and chemokines that recruit more phagocytes to help in battle, inducing more inflammation in the injured site. Proteins in the macrophage's membrane called toll-like receptors bind with bacterial lipoproteins and dimerize to form a protein complex that initiates signaling pathways that activate regulators that induce the expression of cytokines and chemokines proteins. Minutes later, macrophages start releasing IL-23, IL-6 and IL-12. Interleukin-23 binds with receptors on type 3 innate lymphoid cells or ILCs3, and activates them. Minutes later, they start releasing chemokines that only attract neutrophils, and IL-17 and IL-22. Interleukin-17 binds with receptors on fibroblast and endothelial cells, inducing them to produce antimicrobial peptides and chemokines that recruit even more phagocytes to the site of battle. Most bacteria succumb to innate defenses, all but one species. After having adhered to the collagen filaments, the Staphylococcus aureus bacteria began forming a slimy barrier that protects them from all kinds of molecular attacks like complement proteins, degradative enzymes, antimicrobial peptides, immunoglobulins, and it also prevents attachment with phagocyte receptors, inhibiting phagocytosis. The slimy substance, called biofilm, is mainly made from polysaccharide polymers that facilitate attachment, named polysaccharide intercellular adhesin or PIA. PIA polymers are synthesized, transported and modified by a complex of 4 proteins. ICAA and ICAD take molecules of N-acetylglucosamine and synthesize polymers of 20 monomers, which are then transported out of the cell by ICC. ICAC not only transports, but it also covalently attaches several polymers, forming a longer polymers. As the PIA polymers exit the cell, every third or fourth monomer is modified by ICAB, making them positively charged by deacetyl.